Hello and welcome to our presentation on CAM and C4 pathways. Here's an example of a CAM plant. Other examples may include pineapple or cacti. And here is the C4 plants, which include corn, sugarcane, and various types of grasses.
But Spencer, what are CAM and C4 plants?
C4 plants are plants that use the C4 cycles during the dark reaction of photosynthesis. When compared to C3 plants, they have a higher optimal temperature, and unlike C3 plants, photosynthesis occurs even when their stomata are closed. However, the main difference between C3 and C4 plants is that single fixation of carbon dioxide is observed in C3 plants, while double fixation of carbon dioxide is observed in C4 plants. C4 carbon fixation is an elaboration of the C3 carbon fixation and is believed to have evolved more recently. C4 plants are able to overcome the inefficiencies of photorespiration and therefore have a higher net photosynthetic rate. When comparing diagrams of C3 and C4 plants, we can see that in C4 plants, the mesophyll cells are arranged concentrically around the bundle sheath cells, and that the bundle sheath cells in C4 contain chloroplasts, while the ones in C3 plants usually do not.
CAM plants are part of a family called Crossulaceae, and they live in warm, dry regions of the world. These plants use crossulacean acid metabolism, also known as CAM, to minimize photorespiration. The main difference with CAM plants is that light-dependent reactions and CO2 fixation are separated by time. At night, CAM plants open their stomata and fix CO2, which forms C4 molecules, and at day, the C4 molecules release CO2. The main advantage of this is it allows the plants to conserve water, which is important for the environment that they live in.
Now that we know what C4 and CAM plants are, let's get into explaining the C4 and CAM pathways. The key step in the C3 or Calvin cycle is the addition of carbon dioxide to RuBP, but approximately 25% of these reactions by the RuBP carboxylase enzyme instead add oxygen to RuBP, creating a product that cannot be used within the Calvin cycle. This process is called photorespiration. It reduces the efficiency of photosynthesis, potentially reducing photosynthetic output by 25% in C3 plants. In order to bypass photorespiration, C4 plants have developed a mechanism to efficiently deliver CO2 to the RuBP carboxylase enzyme. The first part of the C4 cycle starts in the mesophyll cells. The mesophyll is the inner tissue of a leaf. The term comes from the prefix meso, which means middle or between, and the suffix phill, which means leaf. In the mesophyll cells, the first event of carbon fixation occurs. The C4 plants combine CO2 in the form of bicarbonate to PEP to form oxaloacetate using the enzyme PEP carboxylase. Oxaloacetate is the first stable compound in the pathway, and it has 4 carbons, which is why the pathway is called the C4 cycle. The oxaloacetate is then reduced into malate or malic acid by the reducing agent NADPH2. The malate is pumped into the bundle sheath cells where decarboxylase removes the CO2 and leaves pyruvate or pyruvic acid. The malate is pumped into the bundle sheath cells where decarboxylase removes the CO2 and leaves pyruvate or pyruvic acid. Here in the chloroplasts of the bundle sheath cells, CO2 finally enters the Calvin cycle, and we see the second event of carbon fixation occur. Now, the C4 plant has successfully bypassed photorespiration because the RuBP carboxylase enzyme in the Calvin cycle is protected from oxygen.
But if it is a cycle, how does the pathway get back to where we started?
Well, pyruvate or pyruvic acid, which was made earlier from the malate, diffuses back into the chloroplasts of the mesophyll cells, where it is phosphorylized by ATP and is turned back into the original compound, PEP. Wow, that was a lot of information about the C4 pathway. Sam, could you elaborate more about the CAM pathway for us?
Sure. The main difference between C4 and CAM plants is how they minimize water loss. C4 plants relocate CO2 molecules to minimize photorespiration, while CAM plants minimize photorespiration by choosing when to extract CO2 from their environment. CAM photosynthesis begins at night when the stomata open, allowing CO2 to diffuse into the cytoplasm of the CAM mesophyll cells. In the cells, CO2 reacts with OH- to form HCO3, which is an enzyme known as PEP carboxylase. Then, PEP carboxylase catalyzes PEP to form oxalacetate. Oxalacetate then takes an electron from NADH and is catalyzed by MDH, which is malate dehydrogenase. This is then converted into other organic acids, such as malate. These acids are stored in the vacuoles of the cell until daytime. The next day, the stomata do not open, but these plants can still photosynthesize. The organic acids break down and release CO2 because they have available ATP and NADH from the sunlight. This CO2 then enters the Calvin cycle, which is controlled by rubisco, and makes carbohydrates for the plant. This process allows CAM plants to avoid photorespiration, and it is very water efficient, which is important for the environment that these plants live in. For example, C3 plants lose 97% of the water that is taken up by the roots. Photosynthesis in CAM plants is minimal due to the limited amount of CO2 being fixed at night, but it allows CAM plants to live under their stressful conditions.
Thank you for watching our presentation on CAM and C4 pathways.